Hello and hello everyone, welcome to class 12 biology. In this video we are going to discuss about an infectious and deadly human viral disease called AIDS. So AIDS is short for acquired immunodeficiency syndrome. If you recall, when we discuss about common human diseases, under the viral disease section I told you that we are going to study about AIDS in a separate video. So we are doing this separate video because in your textbook more information is given regarding this particular disease, and from your examination point of view also this particular disease is pretty important because many questions have been asked based upon this particular disease in your previous year question papers. Okay, so AIDS is short for acquired immunodeficiency syndrome. And if you try to draw some little information regarding this particular disease from the name itself, acquired immunodeficiency syndrome means acquired, right, disease that is acquired during a person's lifetime. So AIDS is something which is acquired during a person's lifetime, and it's not congenital. It is not something which is present from birth itself. Okay, so it is acquired. Immunodeficiency means deficiency of immune system, right? Your immune system becomes weakened in this particular disease. And syndrome means it shows a group of symptoms, right? So AIDS or acquired immunodeficiency syndrome means a disease which is acquired during a person's lifetime, and this particular disease is characterized by immunodeficiency and it shows a group of symptoms. Okay, now AIDS is caused by human immunodeficiency virus, or HIV in short. These two abbreviations are pretty important from examination point of view. AIDS, it stands for acquired immunodeficiency syndrome. HIV, it stands for human immunodeficiency virus. AIDS is the name of the disease and HIV is the name of the virus responsible for causing that particular disease. So AIDS is caused by HIV. Right. So HIV is a retrovirus. So retroviruses are those viruses in which the genetic materials are RNA, not DNA. So if you look at the structure of HIV, you can see that these two red strands, they are RNA strands, single-stranded RNA strands, and RNA molecules, single-stranded RNA molecules. These RNA molecules, they are enclosed within a protein capsid, and outside the capsid there is one more layer called as viral envelope, and on the viral envelope you can see these protruding glycoproteins, and these glycoproteins are pretty important. They use that for recognition of their target or attaching themselves to their target cells during infection. Okay, so HIV is a retrovirus, and you should remember this particular structure of HIV. Now, the virus responsible for causing AIDS, that is HIV, is believed to have originated from this part of Africa in 1920s, that is in Kinshasa of Democratic Republic of Congo. And that was the time when the virus, it jumped from chimpanzees into human beings. So it crossed species during 1920s in this part of Africa. But the first case of AIDS was reported in 1981 in gay men in America. Right, the first known case of AIDS was reported in 1981 in gay men in America, and therefore it was initially called GRID. GRID stands for gay-related immunodeficiency disease. Right. Later on, this particular disease, it spread all over the world, irrespective of sexual orientation, right, among people, and the name was changed from GRID to AIDS. Now let us discuss about the transmission of HIV infection. That is how HIV spreads from human to human. In simple terms, we can say that HIV spreads from human to human through exchange of body fluids. Right. In your textbook, 4 modes of transmission of HIV infection is given. They are, first one, sexual contact with infected person. So I told you that HIV is one of STIs, sexually transmitted infections, right? So if you have unprotected sex with an infected person, there is a high probability that you will also get infected by HIV virus. Okay, that's the first one. The second one is transfusion of contaminated blood and blood products. Now, if you require blood transfusion, if you require plasma transfusion, right, due to any condition that you might have, like you may have sickle cell anemia, you may have some kind of bleeding disorder, or you may be suffering from dengue, right? During that time, you will need blood transfusion or plasma transfusion. And if those blood and blood products are contaminated with HIV virus, right, you can also contract that virus along with the contaminated blood and blood products. So that is the second one. In the third case, sharing of infected needles, right? If you are a drug abuser, if you use drugs, those drugs which needs to be taken intravenously, and if you share those needles with an infected person, you can also get HIV virus from that infected needles. And the fourth one is from infected mother to her child through placenta. In this particular case, if a mother is HIV positive, there's a high probability that she can pass HIV virus to her child through placenta or during childbirth. Right. So these are the 4 modes of transmission of HIV infection. By understanding the mode of transmission of HIV infection, we can determine a group of people who are at high risk of getting HIV infection. They are also called as vulnerable or high risk group. So the first one are those individuals who have got multiple sexual partners, maybe those who are involved in sex trade, right? So they are at high risk group, right? If they practice unsafe sex, they can get HIV infection, right? And the second one, drug abusers or drug addicts. So those people who take drugs intravenously, they are also in high risk group if they share infected needles, right? And the third one is individuals who require repeated blood transfusion. If there are individuals who suffer from blood related complications like sickle cell anemia, hemophilia, right, they require repeated blood transfusions, right? And in such situations, if they get contaminated blood, if they receive contaminated blood, they can also suffer from HIV infection. Therefore, they are also in high risk group. And finally, we have children born to HIV infected mother. So if the mother is HIV positive, the children can also get HIV infection through her mother through placenta, right? So they are the children, those children who are born to HIV infected mother, they are also at high risk group. So these are the people who are at high risk group of getting HIV infection. Okay, so how you can't get HIV, right? How you cannot get HIV from? So kissing, right? HIV infection does not, HIV does not spread from human to human through kissing, not through hugging, not through sharing of food, right? Not through insect bites, not from toilet seats, not through bathing together, bathing together, not through sneezes and coughs, and not through sweat. Right, these are important because you have to know how HIV virus it spreads from human to human. Right, these are not the methods through which HIV can spread. Now let us discuss about the events that occurs during HIV infection. What happens within the body of an HIV infected person? How does HIV infection leads to development of AIDS? This diagram explains the events which occurs during HIV infection. Therefore, this diagram is really important. You have to study this and you have to understand it completely. In order to help you understand this particular diagram, I have given you a flow chart over here. If you follow this flow chart, you can understand the diagram, or you can understand the events that occurs during HIV infection. When the virus first enters the human body, it goes and attacks the macrophages, right? The cells which are responsible for body's innate immunity, right? So the HIV virus, it enters the human body and it goes and attacks the macrophages, during which it injects its RNA into the macrophage. In this particular diagram, let's say the square box represents the macrophage, right? And this one, retrovirus, is the HIV virus. So HIV virus, it goes and binds to the cell membrane of the macrophage and it injects its viral RNA into the macrophage. Inside the macrophage, the viral RNA gets copied into viral DNA, right, through a process called reverse transcription, which is aided by enzyme reverse transcriptase, right? So these two words are important: reverse transcription, which is aided by reverse transcriptase. Okay, so viral RNA gets copied into viral DNA through reverse transcription, and the viral DNA then gets incorporated into the genome of the host. Right. So the viral DNA, it goes inside the nucleus of the macrophage and it gets incorporated into the DNA of human being, right? And then this particular cell starts producing new viruses, because now inside this particular DNA, the viral DNA is also present. Now this particular host cell starts producing new viruses. Therefore, this particular macrophage acts like a HIV factory. These new viruses, they go outside and starts infecting other cells, right? They can attack other cells like helper T lymphocytes and other macrophages as well, during which the number of helper T lymphocytes starts reducing because it kills the helper T lymphocytes. Now, when significant number of helper T lymphocytes gets killed, what happens is the person suffers from immunodeficiency or AIDS. Okay, so that is the events that occurs during HIV infection and how HIV infection leads to development of immunodeficiency or AIDS. So what are the symptoms of HIV infection? Right. The virus, when it infects more and more helper T lymphocytes, the patient suffers from bouts of fever, diarrhea, and weight loss. Right. So bouts of fever, diarrhea, and weight loss are the symptoms of HIV infection. So that means HIV is infecting more and more T lymphocytes. Now, when considerable decrease in the number of T lymphocytes is observed, right, the person now suffers from immunodeficiency. Now the person is facing a severe immune weakening, right? The person's immune system is severely weakened. When the person's immune system is severely weakened, the person then becomes susceptible to infections by pathogens such as mycobacterium, viruses, fungi, toxoplasma, etc. So these pathogens, they are said to be opportunistic pathogens because in a healthy person with a healthy immune system, they cannot cause diseases. The healthy immune system can easily take care of these pathogens. But in a person who is suffering from HIV AIDS, right, whose immune system is weakened, these pathogens, they can start causing complications or start causing diseases. Therefore, these pathogens are called as opportunistic pathogens. So in a HIV patient, right, whose immune system is now weakened, these pathogens, they will start causing diseases, right? So the person suffers from infections by these pathogens. To test for HIV infection, a widely used diagnostic test is ELISA. ELISA, short for enzyme-linked immunosorbent assay, right? So this particular assay looks for the presence of antibodies produced against the virus, or it also looks for the presence of viral antigens, right? When a person's body gets infected by HIV virus, right, the person produces antibodies against it. The immune system produces antibodies against it. So the presence of antibodies will indicate the presence of the virus within the body. Okay, so this diagnostic test looks for the presence of antibodies which is produced against the virus, or it directly looks for the presence of viral antigens. There are different types of ELISA. Here I have shown you the example of direct ELISA, in which a surface is coated with a sample. So let's say we take a blood of a person, of a suspected person, then we coat the blood of that particular person onto the surface, and if there is a virus present over there, the virus will also get coated on this particular surface, right? And then we introduce an antibody which is conjugated or which is linked to an enzyme. So you can see the enzyme linked, right? So the enzyme which is linked to our antibody is introduced. This antibody reacts specifically with the virus particle, right? And it will bind over there, right? And when we introduce a substrate, later on when we introduce a substrate, the enzyme which is linked to the antibody reacts with that particular substrate and creates a color change, right? And if the color change is observed, we can say that the person's blood contains the virus, right? If the virus is not there, there is nowhere the antibody will bind to the surface, and there will be no color reaction later on, right? So the color reaction shows that there is a virus right on the surface, right? Therefore the antibody is reacting with that particular virus, attached to the particular virus, and the enzyme which is linked to the antibody has shown that particular reaction. Therefore it proves that the virus is present within the sample. For the treatment of HIV positive patients, antiretroviral therapy or ART is used. So in antiretroviral therapy, the patient is treated with antiretroviral drugs, and this particular treatment is partially effective. These drugs, they can prolong the life of the patient, but they cannot prevent death. So death is inevitable, right? So what antiretroviral therapy or what antiretroviral drugs can do is it can prolong the life of the patient. How does it do that? So let us look at the drugs, right? Antiretroviral drugs. Antiretroviral drugs are those drugs which can prevent the growth of retroviruses. So HIV is a retrovirus, right? So antiretroviral drugs are usually those chemicals which can inhibit the functioning of reverse transcriptase enzyme, right? You remember, reverse transcriptase has got a significant role in the reproduction of HIV in the macrophages, right? So if there is a drug which can inhibit the functioning of reverse transcriptase, it can prevent the growth of HIV within the macrophages, right? So this will prolong the life of the patient, because since now this virus cannot reproduce, it cannot infect other cells, but it cannot prevent death, because if you stop taking antiretroviral drugs, right, the virus, it will start reproducing immediately, right, and then it will start infecting other cells, and it will cause AIDS and cause death, right? So death is inevitable, right? So antiretroviral therapy can prolong the life of the patient. So there is no cure. These antiretroviral drugs, they cannot kill the HIV virus, right? They can just stop the growth of HIV virus, right? But they cannot completely wipe out the virus inside the patient's body. So there is no cure for that. Since there is no cure for AIDS, prevention is said to be the best option. So what are the prevention measures available for AIDS? Right. So the first one is educating or spreading awareness about AIDS. So educating and spreading awareness about AIDS among general population can control the spread of HIV virus. Right. So for infectious diseases like AIDS, they tend to spread faster among population who are ignorant about the disease. Right? If the general public does not know about how this particular disease spreads, what are the health risks associated with this particular disease, among such populations, the disease tends to spread faster. Right? And if you spread awareness, if you educate people about the particular disease, the people will take preventive measures on their own. Right? That will help in preventing the spread of the disease. Right. So there are governmental organizations like NACO, National AIDS Control Organization, under the division of Ministry of Health and Family Welfare, Government of India. And then there are non-governmental organizations as well, which work very hard in order to educate, or in order to prevent, in order to spread awareness about AIDS. Okay. And then there are other measures as well, like making blood, especially from blood banks, safe from HIV. I told you that HIV transmission can happen through contaminated blood during blood transfusion. Right? If you make the blood from blood banks safe from HIV, you can prevent the spread of HIV. Okay, then we can use disposable needles and syringes instead of repeatedly using those needles. Right? I told you that HIV can also spread through infected needles. Right? So if you use disposable needles, that will prevent the spread of HIV. And then we have advocating safe sex and free distribution of condoms. I told you that the spread of HIV can also happen through practice of unsafe sex. If you advocate safe sex and you use condoms, right, if you freely distribute condoms, people will start using condoms, and that will prevent the spread of HIV virus. And then we have controlling drug abuse. Right, so those people who abuse drugs, who use drugs intravenously, they tend to suffer from HIV infection. Right, so if you control drug abuse, you can control the spread of HIV. And then finally, we have regular checkups for HIV in the susceptible populations. By susceptible population, we mean high risk groups. So those people who are more susceptible to getting HIV infection, you should do regular checkups, like people who are involved in sex trade. Right, so people who are involved in sex trade, they are more susceptible to HIV infection than other groups. Right, so in such susceptible population, you should do regular checkups of HIV, right, so that you can control the spread of HIV among the population. So these are some prevention methods available for AIDS. Okay, so with this we have finished our discussions on AIDS. Right, next we will discuss about cancer. Right, please read about cancer, and then you can watch the next video.